Welcome to Dental Board. This is a biochemistry review, glycolysis lecture number 4. Okay, so in our previous lecture, glycolysis 3, we reviewed the most important regulatory pathway or point in biochemistry in terms of glycolysis. Okay, and this was the phosphofructokinase uh, catalyzed reaction. Okay, so we're going to review the remaining two regulatory points. Okay, all right. So the fructose 2,6 bisphosphate. Okay, is a compound which was found in high concentrations in rat livers. Okay, and it maintained these rats were maintained on high glucose diet. Okay, so it was found that this molecule, okay, is a potent effector. Fructose 2,6 bisphosphate is a potent effector of phosphofructokinase. Okay, we have reviewed the activity of phosphofructokinase in the previous lecture in glycolysis lecture 3. Okay, however, so this molecule is not an intermediate of glycolysis. Okay, so glycolysis does not produce fructose 2,6 bisphosphate, and you have to make sure that you know that this fructose 2,6 bisphosphate is a different molecule than or different from fructose 1,6 bisphosphate. Okay, remember, fructose 1,6 bisphosphate is produced through the action of phosphofructokinase. Okay, so phosphofructokinase adds a phosphate to fructose 6 phosphate. Okay, therefore producing fructose 1,6 bisphosphate. All right, so this fructose 1,6 bisphosphate is produced by the action of phosphofructokinase. Okay, now liver fructose 2,6 bisphosphate acts on phosphofructokinase by increasing the affinity for fructose 6 phosphate. Now you have to remember that the fructose 6 phosphate is the substrate for phosphofructokinase. If they ask you what's the substrate for phosphofructokinase, it's fructose 6 phosphate. And the product of a phosphofructokinase reaction is fructose 1,6 bisphosphate. Okay. However, the fructose 2,6 bisphosphate increases the affinity of phosphofructokinase. So it increases the activity of phosphofructokinase. Now, you have to remember that AMP also increases the activity of phosphofructokinase. So we have now we have two things which we know for sure increase the activity of phosphofructokinase. So AMP and fructose 2,6 bisphosphate. They increase activity of phosphofructokinase. Okay? And remember in the last lecture we discussed about that the two substrates or the two molecules that decrease the activity of phosphofructokinase is high ATP and high citrate. Okay. All right. Moving on. So, last lecture we discussed about phosphofructokinase. Now, the other two irreversible steps, okay, in glycolysis, which are key regulatory points, are the reaction of hexokinase. Remember, hexokinase adds a phosphate on glucose initially when it enters the cell, and this forms glucose 6 phosphate. Okay, and fructose 6 phosphate does not leave the cell. Okay, so when the glucose enters the cell, it is acted upon by hexokinase to form 6 glucose 6 phosphate. And glucose 6 phosphate, the difference from the actual glucose is that glucose 6 phosphate traps glucose inside the cell. Okay. Now, the other regulatory point is pyruvate kinase reaction. All right. All right. So, the second control point in glycolysis is the phosphorylation of glucose to form glucose 6 phosphate. I just talked about that. Okay. And this reaction characterized by hexokinase is reversible. So, irreversible reaction are key regulatory points, and it is responsible, as I just mentioned, for trapping glucose inside the cell so that glucose doesn't leave. Remember, glucose diffuses in and out of the cell. Okay, so to prevent it from diffusing out of the cell, the glucose is converted to glucose 6 phosphate. All right. Uh, and this is because the cell membrane is permeable to charged compounds such as sugar phosphates. Okay, so the sugar phosphates are impermeable, so meaning they cannot just diffuse out of the cell. All right, so glucose 6 phosphate is trapped inside the cell. Okay, all right. So in terms of hexokinase, okay, which remember the kinase always adds a phosphate to a molecule. So hexokinase is inhibited by glucose 6 phosphate. Remember, hexokinase reaction. So you have glucose and you have hexokinase. And this reaction needs ATP, remember, okay? And it forms G6P, glucose 6 phosphate. And this glucose 6 phosphate, when it's too much, it goes ahead and blocks this one, okay? It blocks hexokinase, all right? So hexokinase is inhibited by glucose 6 phosphate, all right? Okay, so these are key things that you're supposed to know about glycolysis, all right? So glucose 6 phosphate accumulates in the cell as a result of building up of fructose 6 phosphate when phosphofructokinase is inhibited. All right. So if you inhibit phosphofructokinase, it means the fructose 6 phosphate is not converted to fructose 1,6 bisphosphate. So, and you know, fructose uh, fructose 6 phosphate is converted or is produced from glucose 6 phosphate. All right. So if you block phosphofructokinase, all these other points are going to be blocked. All right. That's why phosphofructokinase is the major important regulatory point in glycolysis. Okay. All right. So the steps that commit glucose to glycolytic pathway is the formation of fructose 1,6 bisphosphate. And this reaction is characterized by, guess what? Phosphofructokinase. Okay. So this this reaction characterized by phosphofructokinase commits glucose to glycolytic pathway. So if they say which of the following enzyme commits glucose to glycolytic pathway, it's phosphofructokinase. All right. And in glycolysis, this important regulatory point is done by, okay, by phosphofructokinase step. That's why you have to memorize or know phosphofructokinase. All right. So the third control point in glycolysis. So the first control point in glycolysis is the phosphofructokinase reaction. The second regulatory point for in glycolytic pathway is the hexokinase reaction. Now the third is the pyruvate kinase reaction. Okay, so pyruvate kinase reaction transfers the phosphate from phosphoenopyruvate. Okay, transfers the phosphate from phosphoenopyruvate PEP to ADP, forming ATP. So this reaction, remember, glycolysis forms a net of 4 ATPs. I mean, a gross of 4 ATPs. However, the net is 2 ATPs. Okay, so the most important portion, remember, the pyruvate kinase reaction, all right, forms ATP. Okay, it forms ATP because the phosphoenopyruvate transfers the phosphate to ADP, forming ATP. All right, and the pyruvate kinase reaction is inhibited by ATP. So when you form too much ATP, then you don't need to produce more. So this reaction is inhibited. Now, so meaning ATP, okay, ATP inhibits pyruvate kinase, okay, and the other thing which inhibits pyruvate kinase is also alanine. So alanine also inhibits pyruvate kinase. So you have to know that, okay? So when the cell's energy charge, remember when the cell's energy charge, meaning the ATP is high, glycolysis is inhibited. When the cell's energy charge is high, glycolysis is inhibited. And also, you have to remember that pyruvate kinase is also stimulated by fructose 1,6 bisphosphate. Stimulated, not inhibited. So the fructose 1,6 bisphosphate stimulates PK, pyruvate kinase. Okay, but ATP and alanine inhibit pyruvate kinase. So keep those things in mind. Okay. All right. So in response to low blood sugar, in response to low blood sugar, glucagon is released and triggers a phosphorylation cascade. All right. And the primary tissues which need glucose are the brain and the muscles. All right. The brain and the muscles need enough glucose. And so the phosphorylation dysphosphorylation control of pyruvate kinase prevents the liver from using glucose when it is needed by other tissues. Okay. So you may feel weak, but the brain is going to need that glucose so you can continue thinking. Okay. Now, pyruvate kinase is also controlled by covalent modification, okay, meaning phosphorylation. So phosphorylation, dysphosphorylation controls the activity of pyruvate kinase, all right? And the phosphorylation of pyruvate kinase makes pyruvate kinase less active, all right? Phosphorylation of pyruvate kinase makes the pyruvate kinase less active, all right? Therefore, dephosphorylation of pyruvate kinase is more active. Remember these 2 points, okay? I like to ask questions about those, all right? Okay, so here's a chart to review. Metabolic condition, enzyme involved, and the effect. So this is a good summary of what we discussed, okay? So if you have low energy charge, meaning low ATP, the phosphofructokinase reaction is stimulated, okay? So glycolysis is stimulated, all right? High energy charge, so that means you have too much ATP. The phosphofructokinase and the pyruvate kinase reactions are inhibited or slowed down. Now, if you have high G6P, glucose 6 phosphate, what's going to happen? G6P, if it's too much G6P, hexokinase is inhibited. So glycolytic pathway is inhibited. Now we talked about citrate. Citrate inhibits the action of phosphofructokinase. So high citrate inhibits phosphofructokinase. So therefore glycolysis is inhibited. So this is very important summary. That you need to know, okay? All right. So, high fructose 2,6 bisphosphate, we talk about in the liver. This fructose 2,6 bisphosphate is not, I repeat, is not produced in glycolysis. However, when it's available, the fructose 2,6 bisphosphate, when it's available, especially in the liver, it increases the activity of phosphofructokinase. So glycolysis is stimulated. If you have high fructose 1,6 bisphosphate, pyruvate kinase is stimulated, so glycolysis is stimulated, right? High alanine, we talked about how high alanine is going to inhibit pyruvate kinase, all right? So it's glycolysis is inhibited. And if you have glucagon, if you have high glucagon, it means you have your sugar is low, so you are going to break down whatever storage glycogen that you have. So pyruvate kinase is inhibited. So high glucagon, low blood sugar, all right, pyruvate is inhibited. Why is pyruvate kinase inhibited? Because you have low blood sugar, and pyruvate kinase is part of glycolysis. So what happens is that you need glucose to undergo glycolysis, because glycolysis, remember, is the breakdown of glucose. Okay, so this is key chart for your review. All right. Okay. So let's a quick review about this statement. So is this statement true or false? Fructose 2,6 bisphosphate is an intermediate in glycolytic pathway. Remember, I just repeated this. So this is false. Okay. So the answer is false. Fructose 2,6 bisphosphate is not an intermediate in glycolytic pathway. Okay. All right. The hexokinase step of glycolysis is inhibited by which of the following? Remember, we say the hexokinase is inhibited by G6P. So the answer is going to be our glucose 6 phosphate, G6P. Okay. All right. Simple review questions. Okay. All right. How about this one? Phosphorylation of the enzyme pyruvate kinase results in what? Phosphorylation. Remember, so you have to know phosphorylation of pyruvate kinase inactivates. Okay, it makes, if you phosphorylate pyruvate kinase, the pyruvate kinase is going to be inactive, so there's going to be less ATP. So phosphorylation, it results in, if you phosphorylate this, you're going to result in inhibition of the kinase reaction or stimulation of glycolysis. No. So glycolysis is not going to be stimulated. All right. So this is out. Release of glucagon. Is it going to release glucagon? Is it going to inhibit glycolysis? Yes, you're going to inhibit glycolysis. So glycolysis is inhibited, right? Because pyruvate is a pyruvate kinase forms the ATP from glycolytic pathway. So if you inhibit this process, the other substrates such as fructose 1,6 bisphosphate or fructose 6 phosphate or glucose 6 phosphate, all those are going to accumulate and then they are going to cause feedback inhibition, okay, of the enzymes. So glycolysis is going to be inhibited. So remember that, okay? All right. So that's the end of our review. Quick and sweet. Okay, very important key points that you're supposed to know. All right. So don't immerse yourself into some other nitty gritty of small enzyme conversion or denaturation of the enzyme. Just make sure that you know the key steps of glycolysis. So review the glycolysis lecture 1, 2, 3, and 4. Okay. All right. So for you to receive notification on additional lectures, subscribe, like, and share. Okay. Good luck and have a nice day.